we are pleased to present ProtoSynth AI. This proof of concept explores how USDM digital protocols, CDISC standards metadata, and AI-assisted automation can be used to generate traceable, standards-aligned synthetic clinical trial data. Before we begin, this application is a proof of concept created for demonstration, research, and evaluation purposes only. This solution was developed through collaboration across CDISC standards expertise, AI and application development, and product leadership. ProtoSynth AI is designed to primarily address three important needs. The application supports standards compliance by leveraging USDM to SDTM traceability and standards metadata. It also enables downstream analytics and automation by providing realistic data earlier for SDTM, ADM, TLF programming, dashboards, AI models, and metadata-driven pipelines. The application also supports study preparedness and test workflows or integrations earlier in the study life cycle. This slide shows the high-level workflow of the application. The first layer provides configurable AI prompts which trigger the LLM model to use steering specifications and CDISC library API key to access machine-readable CDISC resources. Next, the data parsing process begins with a digital protocol in JSON or Excel format. The application parses study metadata such as study design elements, endpoints, and biomedical concepts. In parallel, the standards intelligence layer employs machine-readable data standards and DOG references to generate CDISC-aligned datasets. A machine-readable list of required datasets and variables is generated as intermediate outputs. These are reviewed and approved through a human-in-the-loop step. Finally, the output layer produces synthetic SDTM datasets, along with a compliance and traceability report. The key point is that generation is not random. It is guided by protocol metadata, standards metadata, and documented generation rules. Traceability is a central part of the solution. Each generated SDTM variable is linked back to a source or rule. Here are few examples. The values of gender variable, sex, in demographics dataset are mapped to the values corresponding to USDM population definition class and subject sex attribute. Likewise, laboratory test codes values in lab dataset are mapped from USDM biomedical concepts in main timeline class. And visit start date in subject visit dataset is derived using a synthetic visit schedule rule, leveraging study start date from digital protocol. This creates transparency from protocol metadata and standards guidance to generated SDTM values.
Now let's switch to the live demo and see how ProtoSynth AI converts digital protocol metadata into synthetic, standards-aligned study data. Here is landing page of ProtoSynth AI application. The application has a navigation pane on the left and an action screen on the right. Let's start with the first step, app configuration. Here we have LLM model settings and dataset generation parameters. The application allows us to choose the LLM provider and configure it. The dataset generation parameters like subject count, validation level, randomization seed, and implementation guide version can also be configured here. AI prompt template and CDISC library API settings are also shown here. Next step is to input files. Here let's upload a USDM digital protocol in JSON and Excel formats. These files provide protocol-level metadata. Once the protocol is processed, the application extracts key study characteristics. The intent is to make protocol metadata actionable. Next, we upload a simple text file containing CDISC library API key to access machine-readable standards from CDISC library. Next, we upload the synthetic data generator LLM specifications file. This file provides dataset to USDM mappings. We can view a preview of the contents of this file here. These mappings will enhance accuracy and reproducibility of the mapped information, as well as reduce LLM model hallucination. Next, we provide a path to an internal repository of all available CDISC therapeutic user guides. Finally, we choose the path to output files and select the required format of the output datasets. With these input files, the application applies standards intelligence. USDM elements are mapped to appropriate SDTM concepts, domains, and variables. We can also preview high-level metadata here before we move to the next steps. Now, we move to validating the input sources. In this step, the application verifies the access to input files and parses the information in digital protocol. In the next step, the domains identified are listed for the user to review and choose to keep only the required datasets. Finally, we generate synthetic data. Here we can toggle to the results section where we can first see detailed traceability maps for each dataset and variable. The information is also presented in a table format where traceability paths are shown for each dataset. Next, a preview of synthetic datasets generated is also shown. Traceability maps for each variable can also be found here when you hover over each variable. The validation tab here shows if the dataset passed all standards and metadata conformance checks. We can either download each dataset or click on Download All to download a zip file. In summary, ProtoSynth AI demonstrates how digital protocols can become executable metadata assets for generating traceable standards-aligned synthetic clinical trial data.